Bismillahirrahmanirrahim. 9700 A-Level Biology, March 23, paper 4-2. And this is the third video, and we might be able to finish 7, 8, 9, and 10. Starting with question number 7, this is the third video. Figure 6.1 is a diagram represented a synapse between a chemoreceptor cell from a human taste bud and a dendrite of a sensory neuron. Microvilli, chemoreceptor cell, synapse, dendrite of a sensory neuron. In an experiment, different concentrations of sodium chloride solution were applied to the microvilli of the chemoreceptor cell. The membrane potential of the chemoreceptor cell and the membrane potential of the dendrite of the sensory neuron were recorded for each concentration. The resting potential of this chemoreceptor cell is negative 50 millivolts, and the resting potential of the dendrite of the sensory neuron is minus 70. So this is minus 70 millivolts. You do this working, and the chemoreceptor is minus 50 millivolt. And what is this? This is the resting potential of the dendrite, and this is the resting potential of the chemoreceptor. So negative 50 and negative 70. The results are shown in table 7.1. Concentration of sodium chloride 0.1, chemoreceptor cell minus 50, dendrite minus 70, 1 plus 30, 10 plus 30, dendrite of sensory neuron plus 40 plus 40. So when you place them, the resting potential in an experiment, different concentrations of sodium chloride were applied to the microvilli, and the membrane potential of the chemoreceptor cell and the dendrite and of the sensory neuron were recorded for each concentration. So you can see how in point 1 it was minus 50 and it was minus 70. Then in 1 sodium chloride it went up to plus 30. With 10 sodium chloride went up to plus 30. And similarly in the sensory neuron it went up to plus 40, and in 10 sodium chloride gram per dm cube it also went up to plus 40. So take a little time, look at this table, and then read the next part of the question, which is explain the results shown in table 7.1. So you have to explain the results. With 0.1 gram, chemoreceptor does not release neurotransmitter into the synapse, so is not depolarized. Dendrite of the sensory neuron remains at resting potential. With 1 gram solution, chemoreceptor releases neurotransmitter, membrane potential increases to plus 30. Membrane potential of sensory neuron increases to plus 40. So action potential or depolarization in dendrite of the sensory neuron. And then of course there is a threshold, all or none law in either of the cell. This is the rule that we follow. Now looking at this table, you see because the question said explain the results. Now looking at the table, let's go through it once again. With 0.1 gram, what has happened? Chemoreceptor does not release neurotransmitter. Why is it saying? Because it's not depolarized. It has remained, the resting potential has remained minus 50, minus 50. This has also remained minus 70, minus 70. So with 0.1 gram dm cube, chemoreceptor does not release any neurotransmitter, or you can say it's not depolarized. So dendrite of the sensory neuron remains at resting potential. 0.1 kilogram, the neurotransmitter synapse is not depolarized. Now the sensory neuron, dendrite of the sensory neuron has also not been depolarized. So it remains at the resting potential. With 1 gram, now what has happened? Let's look at 1 gram. Now chemoreceptor releases neurotransmitter because the membrane potential increases to plus 30 millivolts. And membrane potential of the sensory neuron increases to plus 40 millivolts. So action potential in the dendrite of the sensory neuron. So there is an action potential generated in the dendrite of the sensory neuron. And because there must have been a threshold which has been crossed, that is why an action potential has been generated in the dendrite of the sensory neuron. With the 0.1 gram dm cube, chemoreceptor is not depolarized. Dendrite of sensory neuron remains at resting potential. With 1 gram, membrane potential increases to plus 30, or you can say it is depolarized. Membrane potential of sensory neuron increases to plus 40. So depolarization in dendrite of sensory neuron. Threshold has been crossed, or you can have mentioned all or none law in the context of any of these cells. Then B part of the question is describe the differences in structure and function between sensory neurons and motor neurons. A very direct question, nothing very ambiguous. It's very, very clear. Sensory neuron carries impulses from receptor to CNS, while the motor neuron carry impulses from CNS to effector. Cell body of the sensory neuron is between the dendron and the axon. Cell body at end in the motor neuron. Sensory neuron has afferent axon or dendron, and axon. Motor neuron has a long axon. No dendrites from the cell body of the sensory neuron. Dendrites extend from cell body of the motor neuron. So you had to do a direct comparison, this versus this versus this. So you could have drawn even a line in between and written sensory and motor neuron and given me the comparative analysis. So you can see the four comparisons which I've made, and I've made these headings sensory and motor. And you could do the same thing, you could do a comparative because the comparison got you that 1 mark. If you wrote both the things, only then you got 1 mark. Like if you said sensory receptor to CNS and motor CNS to effector, only then you got a mark. Because it says describe the differences in structure and function between sensory neurons and motor neurons. So you could talk about the structure or the function, but you had to do the differences. So they carry impulses from receptor to CNS, that's a function. The CNS to effector, then cell body between dendron and axon, cell body at one end has dendron and axon, has a long axon, and no dendrites from the cell body. In the sensory neuron, there are dendrites extending from the cell body of the motor neuron. So easy question. Another very clear, direct question. Describe the functions of the internal membranes of the chloroplast in photosynthesis. And this is for 7 marks. So it's a long, and one must try and get all these 7 marks because these are very easy, direct questions. So functions of the internal membranes. Now, if you want to do a diagram and do it on that, you can also do that. So there are about, how many points? There are about 12 points, and you could have given me any 7. So you could have said about the location of the light absorbing pigments, then photoactivation leading to emission of the electrons. Then you could have talked of the cyclic and the non-cyclic photophosphorylation, or the light dependent stage. Then you could have said about the site of photolysis, and then location of the carriers of the electron transport chain, then the ATP synthase which is present uh, on the thylakoids, then production of ATP and production of reduced NADP from the Calvin cycle, then chemiosmosis, then the thylakoid space for accumulating the protons, and the stacks to form the grana. And this, of course, gives you a very large surface area for these photosystems to be embedded in it. So one can see a whole lot of mark scheme points for this photosystem 1 and 2 on the thylakoid membrane, light absorbing pigments, site of cyclic and non-cyclic photophosphorylation, site of autolysis, location of carriers of ATC, location of ATP synthase, production of ATP and reduced NADP, chemiosmosis, enclose the thylakoid space for accumulating protons, and stack to form the grana, and gives very large surface area. So about 11 mark scheme points, and you had to give me only 7 to get your 7 out of 7. Rubisco activase RA is an enzyme that has an effect on the activity of Rubisco. An investigation was carried out on the effect of RA on the activity of Rubisco. Solutions of Rubisco and RUBP were added to 2 tubes A and B. So we have 2 tubes A and B, and solutions of Rubisco and RUBP were added. So solutions of Rubisco and RUBP. RA was added to tube A, and RA is what? Rubisco activase. RA was added to tube A. Both tubes were incubated 25 degrees for 6 minutes. The activity of Rubisco was measured over 30 seconds. All conditions were kept the same except the addition of RA to tube A. Here it was not added, and it was added to A. The results are shown. As you can see in tube B, we get a straight line, so Rubisco activity in arbitrary units is sort of negligible. This is the one for B which did not have RA. And for test tube A, it has gone up. Let me give it another color. It's gone up, and the activity is in arbitrary units. So in the beginning also it was more than the one in B, and then it has increased over a time of 6 minutes. Describe the result shown in figure 8.1 and suggest an explanation for the effect of RA on the activity of Rubisco. So this was describe and suggest an explanation. So the describe is always the one which is the easiest, and you can get full marks on that. And I always say that this is something which you can, not knowing any bio, but still you can get marks here. So now when you look at the describe, that was very easy. A activity increases steeply at first and then increases less steeply. Then if you give me a data code, 1 minute for A and 1 minute for B and then 6 minutes for A and B, then RA, which was what? Rubisco activase, changes the active site of Rubisco. Now these red are the explanations. These are the explanations. So Rubisco activase or RA changes the active site of Rubisco. RA enables Rubisco to bind more readily with RUBP, and more enzyme substrate complex is made, enables the product to leave the active site more quickly. So I mean, any enzyme reaction which is occurring faster means that you have to talk about the active site. That means it changes the active site, enables the enzyme substrate complex to form more readily. More enzyme substrate complex is made, more product per unit time. So more product will leave the active site and it will be more product formation per unit time. Question 9, figure 9.1 is a diagram of a relaxed sarcomere in striated muscle. So you can see the Z line, M line, and if you remember my videos, is the dark bands have the A bands and the M line, and the light is the I band and the Z line, and Z to Z is sarcomere. So on figure 9.1, use label lines and letters to label an actin filament with the letter P and a myosin filament with the letter R. So let me just call this, this is the dark one, so this would be the R. Any one of these could be. An actin filament would be the P. So this would be the actin filament. So this would be, you label it with a P. So pointing to the thin filament P and R to the thick filament, and state what happens. Now that was 2 marks. State what happens to the A band and the I band when the sarcomere contracts. So I made you mnemonic for this. H and I become shorter. So the A band will stay the same. A band is not going to change, while the I band will get narrower. So this was again another 2 marks. So it's the H zone and the I band which actually become shorter. But here, of course, they have not shown you the H zone. The plant Stricus toxifera produces the toxin Curare, which can cause muscle paralysis in mammals. The toxin acts by binding to receptors on the cell membrane sarcolemma of the muscle cells at neuromuscular junctions. Suggest how binding of curare to receptors may cause muscle paralysis. So if you know the muscle contraction story, you just have to say, okay, then that will not happen. So number one, this curare will block the receptors. So no sodium ions will enter the muscle fibers. So no depolarization of the sarcolemma. Sarcolemma is the cell membrane of the muscle cells. And no action potential spread across the muscle fibers. So no calcium ions released by the sarcoplasmic reticulum. So no cross bridges will be formed. So basically you have to just say that all those things would not happen which result in a muscle contraction, because what it is doing is it is stopping, because it told you in the question that the toxin curare causes muscle paralysis. So muscles are not going to contract, so no cross bridges are going to be formed. Blocks receptors, no sodium ions enter muscle fiber, no depolarization of sarcolemma, no action potential spread across the muscle fiber, no calcium ion released by the sarcoplasmic reticulum, so no story of the tropomyosin and the troponin and all that, and no cross bridges or a fewer cross bridges will be formed. Coming to the last part of this question, Steph suggests why the action of curare may lead to the death of a mammal. You see, you've got to think of which muscles. If it's the intercostal muscles or the diaphragm muscles or the rib muscles, then the person is going to be unable to breathe. Or if it affects the cardiac muscle, then it stops the heart working. So it will stop blood circulation. So of course that's going to result in the death of a mammal. Affects intercostal muscles, unable to breathe, affects cardiac muscle, stops blood circulation. And now we come to the last question, which is, I think, quite an easy question. The passage in figure 10.1 is about biodiversity. Complete the passage by using the most appropriate scientific terms. Biodiversity within an area can be assessed at different levels, including the species diversity, genetic diversity, and ecological diversity. Species diversity can be assessed by determining the number of different species and the relative abundance, or you can say numbers, or you can say population size. But of course, the best word is abundance of different species in a given area. From this information, species diversity can be estimated using Simpson's index. I think there's a cartoon on that as well. Simpson's index of diversity. Then organisms of the same species can show much genetic diversity even though they share the same genes, or you could have said characteristic traits. This is because they can have different combinations of alleles. The greater the genetic diversity, the greater the ability of a species to adapt to a changing environment or evolve. Ecological diversity is a measure of the number and range of different ecosystems and habitats within a given area. Coming to the B part of the question, the International Union of Conservation of Nature (IUCN) Red List of Threatened Species is updated regularly. Table 10.1 shows the numbers of endangered animal species counted every 3 years between 2007 and 2019. So 2007 number of endangered species 7851, 2010 9618, 2001 3 2013 11212, 2016 12630, and 2019 14234. Calculate the rate of increase in the number of endangered species between 2007 and 2019. So show your working. So this was 7851 and this is 14234. So we want to know the rate of increase in the number of endangered species. Show your working, give your answer to the nearest whole number. This many students do not read and give me the wrong answer. So rate of increase per year is going to be 2 marks. What you have to understand is that from 7,851, the number of endangered species went up to 14,234. So the number of endangered species had increased. So they want you to give us the number, the rate of increase. So this was 2019 minus 2007 would be 12 years. So this is a record of 12 years from 2007 to 2019. So we have to divide that 14,234 minus 7,851 divided by 12, and the nearest whole number is 531.9 or something. So you gave the answer to 532. Then coming to the next part of the question, which is the last part of the paper. More species of fish were listed as endangered in 2019 than species of mammals. Suggest reasons why more fish species than mammal species are endangered. But what is very basic, because you see we've been overfishing. So overfishing regulations, you see it's difficult at sea to enforce protective laws. Then climate change, resulting in lots of oil spills or some even the pollutions will of course also be added. Climate change would mean what? We'd have to talk about what climate change are you talking about? And then pollution, and then there are more species of fish than those there are species of mammals. Mammals, the public is more aware, leading to conservative actions, while they're not aware about the fish. And there is law in place banning hunting, like of elephants and all that. So that is why poor fish are becoming more extinct and endangered and becoming extinct. So any of these points would have been valid for the 4 marks. Overfishing at sea, difficult to enforce protective laws. Like, the enforcement of mesh size is one of the most important thing, but it's very difficult to enforce these laws in these large oceans. Pollution like plastics at sea, more species of fish other than species of mammals. People are more aware of mammals, leading to conservative actions. Laws in place banning hunting, climate change like global warming causing shortage of food.